DNA replication is the process of DNA making a copy of itself during cell division. What better way to explain DNA replication in prokaryotes than with candy? Let's start the discussion by introducing the key proteins that aid in the replication process and make replication possible. Helicase unwinds the double-stranded DNA into single-stranded DNA, forming a replication fork. Single-stranded binding proteins keep these unwound DNA strands single-stranded by coating them in a protective barrier. Gyrase relieves the tension in the double-stranded DNA ahead of the replication fork and prevents supercoiling from occurring. DNA polymerase III is responsible for adding nucleotides to the growing strands of replicated DNA. It adds nucleotides in the 5' to 3' direction, but it is able to proofread its work by working backwards in the 3' to 5' direction. This is known as exonuclease activity. Primase synthesizes primers that allow DNA replication to begin at the origin of replication. DNA polymerase III requires a 3' OH group to attach to in order to add nucleotides. The primers that Primase lays down on the template strand provide the 3' OH that is required to begin replication. These primers are represented by this piece of candy. DNA polymerase I removes the primers through its 5' to 3' exonuclease activity. It then replaces the RNA nucleotides with DNA nucleotides on the lagging strand. Ligase seals the nicks that are formed between the Okazaki fragments of DNA formed on the lagging strand by forming a phosphodiester bond between the two consecutive fragments. Let's now see how these pieces work together in the replication of DNA. At the start of replication, the double-stranded DNA separates into single-stranded DNA at the origin of replication. Helicase continues to unwind the DNA as it continues along the strand in each direction. Primase is also present and will begin to lay down primers for DNA synthesis to begin. Gyrase is found at the beginning of the replication forks, preventing supercoiling from occurring. Because the same process occurs on either side of the replication origin, we can zoom in and focus on just the right side. As we can see, gyrase is still at the beginning of the replication fork. Helicase is unwinding the double-stranded DNA, and primase is laying down primers as the DNA is unwound. We can see the single-stranded binding proteins encasing the single-stranded DNA, ensuring that it stays single-stranded. As we can see, the top red strand represents the leading template strand. The blue strand represents the newly synthesized coding strand. On the top, DNA polymerase III is continuously adding nucleotides in the 5' to 3' direction, making it the leading coding strand. On the bottom, we can see that DNA polymerase III is discontinuously adding nucleotides in fragments as the fork continues to open. Ligase is there to add a phosphodiester bond between the two Okazaki fragments, sealing the nick. DNA polymerase I can be seen removing primers on the lagging strand, allowing for replication to continue. If DNA polymerase I could no longer perform its 5' to 3' exonuclease function, the primers that were laid down by Primase would not be removed from the lagging strand, and RNA nucleotides could not be synthesized into DNA nucleotides. DNA replication could not occur on the lagging strand. I hope you enjoyed this tasty explanation of DNA replication.